Hello friends. Today we'll start with principle of vaccination. Immunity is the ability of a human body to tolerate the presence of material indigenous to the body, that is self, and to eliminate foreign or non-self material. This ability provides protection from infectious disease. Since most of the microbes are identified as foreign by the immune system, immunity to a microbe is usually indicated by the presence of antibody to that organism. Immunity is generally specific to a single organism or the group of closely related organisms. There are two basic mechanisms for acquiring immunity: active or passive. Active immunity is the protection that is produced by the person's own immune system. This type of immunity usually lasts for many years, often during a lifetime. Passive immunity is the protection by products produced by animal or human and transferred to another human, usually by injection. Passive immunity often provides protection, but this protection disappears with time, usually within a few weeks or months. Antigen. The immune system is a complex system of interacting cells whose primary purpose is to identify foreign substance referred to as antigens. Antigen can be either live, such as virus or bacteria, or inactivated. The immune system develops a defense against antigen. This defense is known as immune response and usually involves the production of protein molecules by B lymphocytes, which is called antibodies or immunoglobulins, and specific cells including T lymphocytes, which are also known as cell mediated immunity, whose purpose is to facilitate the elimination of foreign substance. Passive immunity is the transfer of antibody produced by one human or other animal to another. Passive immunity provides protection against some infection, but this protection is temporarily. The antibodies will degrade during a period of weeks to months, and the recipient will no longer be protected. The most common form of passive immunity is that which an infant receives from its mother. Antibodies are transported across the placenta during the first 2 months of pregnancies. In addition to the blood products used for transfusion, that is the whole blood or red cells or platelets, there are three major sources of antibodies used in human medicine. These are homologous pooled human antibodies, homologous human hyperimmune globulins, and heterologous hyperimmune serums. Classification of vaccines. There are basic two types of vaccines: live attenuated and inactivated. The characteristic of live and inactivated vaccines are different, and these characteristics determine how these vaccines are used. Live attenuated vaccines are produced by modifying a disease causing, which we called a wild virus or bacteria, in a lab. The resulting vaccine organism retains the ability to replicate or to grow and produce immunity, but usually it does not cause illness. The general rule of vaccination implies the more a vaccine is similar to the disease causing form of organism, the better the immune response of the vaccine. Now, there are various kinds of vaccines: live attenuated and inactivated form. Live attenuated vaccines are derived from wild or disease causing virus or bacteria. These wild virus or bacteria are attenuated, that is weakened in a laboratory, usually by repeated culturing. I'll give an example. Measles virus used as a vaccine today was isolated from a child with measles disease in 1954. Almost 10 years of serial passage during tissue culture media, this process was repeated to transfer the wild virus into live attenuated inactivated vaccines. These are produced by growing a bacteria or virus in culture media. Then inactivating them, inactivation can be done by heat or chemicals such as formalin. In case of fractional vaccine, organism is further treated to purify only those components which are included in vaccine. For example, polysaccharide capsules of pneumococcus. Inactivated vaccines are not alive and cannot replicate. The entire dose of antigen is administered in the injections. These vaccines cannot cause disease from infections. Even in self or even in the immunodeficient person, that is person that has some deficiency in its immune system. Inactivated antigens are less affected by circulatory antibodies than those of live agents. Inactivated vaccines always require multiple doses. In general, dose does not produce protective immunity but primes the immune system. It activates your immune system. A protective immune response develops after the second or third dose. In contrast to live vaccines, in which the immune response closely resembles natural infection, the immune response to an inactivated vaccine is mostly humoral. Little or no cellular immunity results. Among the various types, next is recombinant vaccines. Vaccine antigen may also be produced by genetic engineering technology. These products are sometimes referred to as recombinant vaccines. Five genetically engineered vaccines are currently available: hepatitis B, human papilloma virus, that is HPV, and influenza. Vaccines are produced by insertion of a segment of a respective viral gene into the gene of yeast cell or virus. The modified yeast cell or the virus produced by the pure hepatitis B surface antigen, HPV capsid proteins, or influenza heme agglutinin when it grows. Live typhoid vaccine, that is TY21A, is Salmonella typhi bacteria that has been genetically modified to not cause illness. That is, if given, it won't cause any illness. Live attenuated influenza vaccines has been engineered to replicate effectively in mucosa of nasopharynx but not in lungs. These are the reference from this for this material. In next session, we'll go in detail about the recombinant vaccine.